Dear students, in this section we will learn about theory of enzyme catalysis, that is Michaelis-Menten theory, then about cofactors and coenzymes, then enzyme inhibitors and uses of enzymes. Please take this section in your textbook, theory of enzyme catalysis, that is Michaelis-Menten theory. So Michaelis-Menten theory or the hypothesis is a general explanation for the velocity and gross mechanism of enzyme catalyzed reactions, and this hypothesis was put forward by Michaelis and Menten in 1913. And let us see the assumptions of this theory. So this theory, first of all, it assumes the rapid reversible formation of a complex between an enzyme and its substrate. Okay, the first assumption is that there will be a rapid and reversible complex formation between the enzyme and the substrate. Substrate means the substance upon which it acts to form a product. The substrate means the substance on which the enzyme will act and will be converted into the product. It also assumes that the rate of formation of the product is proportional to the concentration of the complex. So this is the second point. Okay, that is the rate of formation of this product from this enzyme substrate complex. The rate of formation of this product, that is the speed of the reaction, is proportional to the concentration of the complex, that is the enzyme substrate complex. If the concentration of this enzyme substrate complex is greater, the speed with which the product will be formed will also be higher. The velocity of such a reaction is greatest when all the sites, that is the active sites, at which the catalytic activity can take place on the enzyme molecules are filled with substrate, that is when the substrate concentration is very high. So when all the active sites, when most of the active sites over the enzymes are filled, then the catalytic activity is very high. The velocity with which the enzyme substrate complex is being converted into the product will be very high. So when the substrate concentration is very high, more number of enzyme substrate complexes will be formed, and from that greater product formation will be occurring. So this is the main assumption of this Michaelis-Menten hypothesis. We can see a graph here. This is the graphical representation of this theory. Here you can see that in the y-axis we are plotting the reaction velocity, and in the x-axis we are plotting the substrate concentration. Okay, so you can see an S in square bracket. This represents the concentration of S, that is concentration of the substrate. You can see that as the substrate concentration is increasing, the rate or like the velocity of the reaction is increasing, gradually increasing, and again it is increasing, and at higher substrate concentration it reaches a saturation point. Further increase of velocity is not possible. Okay, so this is a graphical representation of this Michaelis-Menten theory. Now we can see a few terms like Vmax, Vmax half, Km, etc. Let us see what these terms are. Okay. If the velocity of an enzymatic reaction is represented graphically as a function of the substrate concentration S, the curve obtained in most case is a hyperbola. That is what you saw previously. Okay, the graph is like a hyperbola. And the mathematical expression of this curve shown in the equation below was developed in 1912 to 13 by the German biochemist Michels and Menten. And in the equation, V max is the maximal velocity of the reaction and Km is called the Michels constant. So this is the mathematical expression for this hypothesis. So here you can see a V max, which is the maximal velocity of the reaction, and this S is of course the concentration of the substrate, and Km is the Michels constant. So the graph showing substrate concentration versus reaction velocity can be seen here. So here at this point we say that the reaction is at maximum velocity. Okay, through this y-axis, this is the highest portion of the velocity. So here the graph corresponding to this portion, at this region the reaction will be at maximum velocity, and at around half of this portion we say it is half of the maximum velocity, that is V max half. Okay, and this is the Km, that is the Michels-Menten constant. So you have to learn this equation. Here we can see few other terms also. V0. Okay, what is this V0? V0 is the initial velocity. It is the initial velocity V0, that is velocity at time t is equal to 0, that is V0. And V max is the maximal velocity, and S represents the substrate concentration, and here Km, as we said, it represents the Michels-Menten constant. From this graphical representation we can define Km as the substrate concentration at which the enzyme catalyzed reactions attain half maximal velocity. So this is the definition for Km. Km, that is the Michels-Menten constant. So define Michels-Menten constant. Michels-Menten constant, which is represented using the term Km, is actually the substrate concentration at which the enzyme catalyzed reaction attains half of the maximal velocity, that is at V max divided by 2. At V max divided by 2, what is the substrate concentration at this particular point? So this is the half of the maximal velocity. At this point, the corresponding substrate concentration that we get from the x-axis. Okay, that is the value of Km. So Km is defined as the substrate concentration at which the enzyme catalyzed reaction attains half maximal velocity. So next section is enzyme inhibition. So any enzyme inhibitor can bind to an enzyme, and this inhibitor can affect the enzyme in many ways, can affect the action of the enzyme in many ways. So let us see the various type of enzyme inhibitors in this section. First of all, we have to understand that an enzyme inhibitor is a molecule that binds to an enzyme and decreases its activity. So if this enzyme inhibitor, it can be any molecule. Okay, if this is binding to the enzyme, this will decrease the enzyme's activity. The binding of an inhibitor can stop a substrate from entering the enzyme's active site or hinder the enzyme from catalyzing its reaction. So why is this inhibitor molecules decreasing the activity of the enzyme? Because this inhibitor is actually binding to the active site of the enzyme or any other adjacent site. So as a result, the enzyme will not have the capability to act freely, act efficiently for catalyzing a particular reaction. So binding of an inhibitor can stop a substrate from entering the enzyme's active site, or it hinders the enzyme from catalyzing its reaction. Inhibitor binding is either reversible or irreversible. So this is a very important point. So the inhibitor molecules can bind to an enzyme either permanently or non-permanently. Okay. If it is reversible, that is, if this binding can be broken and converted back to the enzyme and the inhibitor separated, then it means that the binding is reversible. But if the binding between the inhibitor and the enzyme is permanent, it means that it is irreversible in nature. It cannot be reversed. The enzyme cannot be freed from the binding of the inhibitor molecule. Then irreversible inhibitors usually interact with the enzyme and change it chemically. Example, covalent bond formation. Okay, if a covalent bond formation is occurring between the inhibitor and an enzyme, this inhibitor is actually changing the chemical constituent or the binding properties of the enzyme. So it is being binded permanently to the enzyme. So it changes it chemically. And these inhibitors modify key amino acid residues needed for enzymatic activity. So we know that most of the enzymes are proteins, and there will be amino acid residues on the surface of these enzymes or at the active sites of these enzymes. So when the inhibitor molecules bind permanently to this enzyme surface, and when it chemically modifies this amino acid residues, then the enzymatic activity will be lost from the active site of that enzyme. So in contrast, reversible inhibitors bind non-covalently, and different types of inhibition are produced depending on whether these inhibitors bind to the enzyme and the enzyme substrate complexes or the both. So here the most important point is that irreversible inhibitors are binding through the mode of covalent bond formation, whereas reversible inhibitors bind through non-covalent bond formation. Now let us see the types of enzyme inhibition. So there are different types of enzyme inhibition. The activity of many enzymes can be initiated by the binding of specific small molecules or ions, and this can be either reversible or irreversible inhibition. First is the reversible inhibition. Okay, so this type of inhibition, in this type of mechanism, the inhibitors attach to enzymes with non-covalent interactions such as hydrogen bonds, hydrophobic interactions and ionic bonds. So all these are examples of non-covalent interactions. Which are they? Hydrogen bonding, hydrophobic interactions and through ionic bonds. So for irreversible inhibition, the inhibitors attach to the enzyme through all these type of non-covalent interactions. Then reversible inhibitors generally do not undergo chemical reaction when bound to the enzyme and can be easily removed by dilution or dialysis. Why? Because the bonding between the inhibitor and the enzyme is not permanent. Therefore, they can be easily removed through the processes like dilution or dialysis. Now, this reversible inhibition, there are different types of reversible inhibitions. So, they are further classified into, the first one is the competitive inhibition. So, what is this competitive inhibition? So, in this type of inhibition, the inhibitor competes with the substrate for the active site of the enzyme. So you can see diagram here, a picture here. So here you can see an enzyme, then a substrate is present, and an inhibitor is also present. So the binding site of the substrate and the inhibitor is similar. So this is a portion which binds to the active site. So here is the active site of the enzyme. So at the active site of the enzyme, either the substrate or the inhibitor will come and bind. But there is a competition between the substrate and the inhibitor for reaching this active site, for occupying this active site. Therefore, what happens is that usually the inhibitor will win. Okay, so the inhibitor will come and occupy this active site. As a result, the substrate will not be able to occupy this site. So here in this type of inhibition, the inhibitor competes with the substrate for the active site of the enzyme. So as a result, enzyme inhibitor complex will be formed instead of enzyme substrate complex. So if an enzyme inhibitor complex is formed, ultimately the enzymatic action of the enzyme will be lost. So enzyme cannot further catalyze any biochemical reaction. So this inhibitor binds to the active site and there will be no biochemical reaction occurring inside the system. So it directly blocks the active site. The inhibitor directly blocks the active site. This type of inhibition is called competitive inhibition. Now the second one is the uncompetitive inhibition. So in this type of inhibition, an uncompetitive inhibitor binds at a site distinct from the substrate active site and binds only to the enzyme substrate complex. So in this case you can see that at first the substrate as usual will come and bind to the active site of the enzyme. Only after that the inhibitor molecule is coming and occupying the second site, an adjacent site or a site distant from the substrate's active site. So here there is no competition between the substrate and the inhibitor molecule. Okay, so this is the final product. There is an enzyme, there is a substrate sitting here, and a different site there is an inhibitor also. So this is uncompetitive inhibition. And again, you can see an expression representing this uncompetitive inhibition. At first, the enzyme and the substrate will form an enzyme-substrate complex. Now, either this enzyme-substrate complex will be converted to the product, or what happens if an inhibitor molecule comes, enzyme-substrate inhibitor complex will be formed. So, this is the representation of this uncompetitive inhibition. Now let us see a third type of inhibition, that is non-competitive inhibition. So in this non-competitive inhibition, inhibitor binds to parts of the enzyme other than the active site. The inhibitor can bind to the free enzyme and also to enzyme substrate complex. So this is also a non-competitive inhibition. Here also the inhibitor is binding to another site apart from the active site. So at the active site the substrate will go and bind. But even before the enzyme substrate complex formation, this inhibitor can directly go and bind to any one part of the enzyme. The inhibitor can bind to the free enzyme, and it can also bind to the enzyme substrate complex to form enzyme inhibitor complex. So this is a type of non-competitive inhibition. Now again, in a fourth type, mixed inhibition. So in a mixed inhibition, in this type the inhibitor can bind to the enzyme at the same time as the enzyme's substrate. Okay, so however, the binding of the inhibitor affects the binding of the substrate and vice versa. So this is mixed inhibition. So in this mixed inhibition, the inhibitor molecule can come and bind to the enzyme, or it can come and bind to the substrate which is attached to the enzyme. So either case can occur, but the binding of this inhibitor is affecting the binding of the substrate, or the binding of the substrate will affect the binding of the inhibitor. Okay, so it is vice versa. So this is a mixed inhibition. So all these four types of inhibitions were a reversible inhibition. Okay, different types of reversible inhibitions. So now we come to irreversible inhibition. So as we said earlier, irreversible inhibition involves covalent bond formation. The bonding will be mostly permanent in nature. As a result, the reversibility of the reaction is prohibited. So in an irreversible inhibitor, this type of inhibitors dissociates very slowly from its target enzyme because it has become tightly bound to the enzyme, either covalently or non-covalently, mostly through covalent type of interaction. So the binding between the inhibitor and the enzyme is very tight, so tightly bounded. As a result, the dissociation between this enzyme and the inhibitor is very slow in nature. So some irreversible inhibitors are potent drugs. Example, penicillin. Okay, penicillin acts by covalently modifying the enzyme transpeptidase, thereby preventing the synthesis of bacterial cell walls and thus killing the bacteria. So penicillin, its action is through the covalent interaction. It covalently modifies the enzyme transpeptidase. So as a result, what happens? This prevents the synthesis of bacterial cell walls. So transpeptidase is present in the bacterial cell walls, on the bacteria. So as a result of the binding of penicillin, bacteria is not able to synthesize its cell walls. Therefore, it results in the killing of the bacteria. Then another potent drug is the aspirin. Aspirin also acts by covalently modifying the enzyme cyclooxygenase and reducing the synthesis of inflammatory signals. So these two are examples for irreversible inhibitions. So irreversible inhibitions, mostly the dissociation between the inhibitor and the enzyme is very slow because both these are tightly bound, either covalently or through non-covalently. Irreversible inhibition can occur. Most common example is how penicillin is acting or modifying the enzyme transpeptidase. Penicillin is a well-known drug and it will modify the transpeptidase chemically, thereby preventing the synthesis of bacterial cell walls and thus killing the bacteria. So the next section is a very important section, that is cofactors and coenzymes. So we know that most of the enzymes are simple globular proteins. Okay, proteins means it is composed of amino acids. Most of the enzymes are simple globular proteins. Some others are conjugated proteins having a non-protein component. So in some of the enzymes, they are of conjugated proteins. Okay, they are composed of conjugated proteins and they have a non-protein component, that is a component which is not a protein, that is which is not made up of an amino acid. So these enzymes require the presence of a non-protein component to be catalytically active. Okay, So what these enzymes require as a must? It requires the presence of a non-protein component in order for it to be catalytically active. So this non-protein part is called a cofactor. So what is meant by a cofactor? A cofactor is the non-protein part of the enzyme which gives it its catalytic activity. Okay, and the cofactor may be a metal ion or an organic molecule. So if this non-protein part is an organic molecule, we call it as a coenzyme. So this is the definition for a coenzyme. If this non-protein part of the enzyme is actually an organic molecule, it can be either a metal ion or an organic molecule. But if it is an organic molecule, we say that it is a coenzyme. It is called a coenzyme. And the protein part in the enzyme is called the apoenzyme. Okay, the spellings are all important. So you have to learn this. What is the difference between a coenzyme and an apoenzyme? Okay, so the non-protein part, if it is an organic molecule, it is called a coenzyme, and the protein part in the enzyme is called the apoenzyme. So apoenzyme combines with the cofactor or coenzyme, forming a complete enzyme. So this is actually an enzyme. An enzyme is composed of an apoenzyme which is conjugated or which is attached to a cofactor or a coenzyme. A complete catalytically active enzyme together with its cofactor is called a haloenzyme. So there are so many different terms here. Okay, so you learned about this cofactor, then coenzyme is there, then you came to apoenzyme. Then now you have the term haloenzyme. So a complete catalytically active enzyme together with its cofactor is called a haloenzyme. So on removing the cofactor, what happens? The enzyme becomes inactive. That is, the apoenzyme is catalytically inactive. You can see this expression here. That is, an apoenzyme, when it is combined with a cofactor and a coenzyme, it is called a haloenzyme. A haloenzyme means it is a complete catalytically active enzyme. And if you are removing this cofactor or coenzyme, the enzyme will become catalytically inactive. The activity of this haloenzyme will be lost. And this is catalytically inactive. So the remaining portion is called the apoenzyme. Now, please see this picture. You can see an apoenzyme. An apoenzyme is a protein portion and this is inactive. And when a cofactor, that is a non-protein portion, comes, or a cofactor or a coenzyme, we use both the terms. Okay, when it comes and binds the apoenzyme, this will become active. So now you can get this haloenzyme. Or haloenzyme means it is an enzyme with a cofactor, isn't it? So there is an active part, a portion that is rendering the activity to this enzymatic part. So this is the haloenzyme, the whole enzyme which is active. So only if this haloenzyme is present here, the substrate will be able to come and attach very efficiently to this active site. Okay, so this is the whole structure of this apoenzyme, coenzyme and the haloenzyme. Now, if this cofactor is any metal ion, then we call it as a metalloenzyme. Okay, so if the enzyme requires a metal ion, then it is called a metalloenzyme. So the metal ion may serve as a primary catalytic center or a bridging group to bind the substrate and enzyme through the formation of a coordination complex, or it may also act as a stabilizing agent. And stabilizing agent, the conformation of the enzyme protein is in its catalytically active form. So these three roles are played by this metal ions. It may either act as a catalytic center itself. Okay, so the binding of the substrate molecule will be over this metal ion itself, or it may act as a bridging group to bind the substrate and the enzyme through the formation of a coordination complex, or it may act as a stabilizing agent. That is, the conformation of this particular enzyme will be stabilized by the presence of this metal ion, so that the substrate can efficiently bind, or the catalytic activity of the enzyme will be maintained. So these three roles are there for the metal ion. So when a cofactor is covalently linked to the molecule, it is called the prosthetic group. So what do you understand by a prosthetic group? So when the cofactor is covalently linked to the enzyme molecule, we say that it is a prosthetic group of the enzyme. So examples are Fe3 plus ion. So this is a cofactor of cytochrome oxidase. So cytochrome oxidase enzyme has a metal ion Fe3 plus, that is a ferric ion. Again, another enzyme called alcohol dehydrogenase, it has a metal ion Zinc 2 plus. Again, you have another enzyme urease. So urease, Ni2 plus is the cofactor of urease. And again another enzyme is there, that is glutamine mutase. Here the cofactor is cobalt, CO2 plus ion. And of course, you know, ferridoxine. So as the name indicates, we can understand which metal ion is present in this enzyme, that is Fe2 plus ion. Okay, ferridoxine is having the cofactor Fe2 plus. So please learn this, which all metal ions are the cofactors of these enzymes. Then some enzymes are synthesized in the organism in an inactive form. This is known as zymogen. Okay, so define a zymogen. What is meant by a zymogen? So in some enzymes are synthesized in the organism in an inactive form. So inside the organism, the enzyme exists in the inactive form, that is, it is not able to bind with any substrate, or it is not able to catalyze any biochemical reaction. Such inactive form of an enzyme is called the zymogen. Example, pepsin. Pepsin is synthesized as its zymogen pepsinogen. Okay. So this zymogen form of this enzyme pepsin is called the pepsinogen. This is the inactive form of pepsin, and it is converted to pepsin in the presence of hydrochloric acid. Okay, so only in the presence of this hydrochloric acid, this pepsinogen will be activated to form pepsin. Now let us see the uses or the applications of enzyme. First application, enzyme is essential for life. So inside the living organisms there are different kinds of enzymes which are having different functions. So without several thousands of enzymes synthesized within the body cells, the chemical and biochemical reactions in the body would not take place, and there will be no life. So these reactions occur with great accuracy and speed due to the catalytic activity of enzymes. So if a person is born without even one essential enzyme, then he may suffer from permanent illness, disability, or even death. So the first first importance is that it is essential for our life. Even the slightest imbalance or the absence of any single enzyme will cause a permanent illness of our body or a disability or even death. And second is the medical applications. Some enzymes are directly used as medicines. For example, streptokinase dissolves blood clot and hence it is used to prevent heart attacks caused by clotting of blood. So this is one of the medical application of the enzyme. Okay, then we have clinical application. The third application is clinical applications. Some enzymes are tissue specific. They will act only on reactions in the tissues in which they are synthesized. Okay, so these enzymes can be synthesized over the tissues in the for the clinical application. So they are tissue specific. So some enzymes are tissue specific and they will act only on reactions in the tissues in which they are synthesized. Because of this tissue specificity, enzymes are helpful in diagnosis of diseases. So this is one of the important application of the enzyme. So enzymes that are synthesized over certain tissues, they exhibit the property of tissue specificity. So only those reactions that are conducted over this tissues can be catalyzed by this enzymes. So this is helpful for diagnosis of certain diseases. When the body tissue is damaged by disease, the enzyme of these tissues are released into the bloodstream. And by analyzing these enzymes, the organ that has been damaged can be identified. So I hope you have heard about biomarkers. Okay, the presence of certain biomarkers in our body. So all these biomarkers indicates the presence of certain biochemical reactions or certain diseases in our body. So if a particular body tissue is damaged by a disease, then the enzymes of these tissues will be released to the bloodstream. So if we have any biochemical sensor, okay, if we have a biosensor that can sense these enzymes, then we can say that because of the presence of these enzymes in the body, this particular organ have undergone certain damage or some diseases. Okay, we can able to sense the proper functioning or dysfunctioning of any particular organ in our body. Then the fourth application is industrial applications. So enzymes are used in pharmaceutical industry for manufacture of drugs like semi-synthetic penicillins and prednisone. Okay, so all these are the synthetic applications of enzymes. So enzymes can be artificially manufactured in the laboratory for large-scale industrial applications. Let us see a few. That is, dry cleaners use enzymes to remove blood, beer, coffee and egg stains from cloth. Then enzymes are used in the processing and tanning of raw animal hide into leather. This is the second application. So for processing of this leather materials from the skin of animal hide, enzymes can be applied. Then enzymes are used in the production of food and drinks by fermentation process. For the manufacture of wine, products like vinegar, wine, beer, bread, cheese, curd, etc. are produced by using yeast and bacterial enzymes. So these are the application in the food industry. Then products like ethanol, acetic acid, lactic acid, citric acid, etc. are also produced by fermentation processes using enzymes. So these are the industrial applications of enzymes. So you can expect questions like discuss the industrial applications or the clinical applications of enzymes. So or you can expect an essay questions like elaborately discuss the applications of enzyme along with the the inhibition, how enzyme inhibition is carried, what are the various inhibition processes of enzymes. Okay, so please learn all those. Then fifth application is analytical applications. Urease is used in the estimation of urea. So the enzyme glucose oxidase is used to detect and measure the amount of glucose in blood using a biosensor. So all these are analytical applications. So urease enzyme is used for the estimation of urea. And another enzyme, glucose oxidase, it is used to measure the amount of glucose present in the blood by using a biosensor. So in the biosensor, we use this glucose oxidase enzyme. Then you have a sixth application, that is organic synthesis. So in organic synthesis, enzymes are used to carry out oxidation reaction, hydroxylation reaction, hydrolysis, reduction and isomerization. All these are different types of chemical reactions. So enzymes can be used to carry out all these different types of chemical reactions. This is because of their easy availability, good catalytic properties, mild reaction conditions, and absence of undesirable side reactions. So here in the field of organic synthesis also, enzymes have many different applications, that to conduct various types of chemical reactions like oxidation, reduction, hydrolysis, hydroxylation, isomerization, etc. So this is the application in the field of organic synthesis. Then a latest application is given, that is the development of environmental friendly, biodegradable plastics and bioethanol by enzymatic conversion of plant starch, that is from the biomass. We are able to produce environmental friendly products like biodegradable plastics or bioethanol, which is used as fuel. Okay, so for all these applications enzymes are used for all these applications. So this is one of the latest application of the enzyme. Okay, so please learn this point also. So it is used for the preparation or for the development of environmental friendly, biodegradable plastics and bioethanol by enzymatic conversion of plant starch. So the material derived from plant product, that is the material starch, is subjected to a certain enzymatic action, and from that products like these are synthesized in the industry.